A variety of computational tools, data resources have been developed by Prime and are accessible via the Prime portal. The Prime database automatically downloads all Prime data nightly from the Prime electronic lab notebook system and provides access to more than 1,000 experimental datasets per Prime investigators, and to most of these datasets, which are completely available after an embargo of 12 months, to the research public. Primed is a database available via the web portal that integrates microarray data from Prime experiments and from other laboratories in a publicly accessible form in order to facilitate the investigation of the antiviral response in dendritic cells. TIDAL is a web-based method for inferring transcriptional networks from dynamic microarray datasets. COREX, or Co-Regulation Explorer, is a web-based tool to analyze and to visualize the correlation between gene or protein expression data and integrate the results from transcription factor enrichment analysis. We also host a table of rate constants useful for developing models of dendritic cells. Part of the rate constant database is shown here. Misty Mountain clustering is a fast, unsupervised algorithm for flow cytometry gating that is available both as a downloadable executable or as a web server-based interface. Placa is a web-accessible tool for inferring functional networks from early gene expression data associated with inhibition experiments of the cell network by RNAi or chemical inhibitors. Biopathways Publisher is a web-based tool for hosting knowledge-based pathway databases, and Prime has developed an extensive database of dendritic cell signaling that can be accessed via this web portal and the Biopathway Publisher tool. And finally, reagents, for example, the monoclonal antibodies that have been developed by Prime are available via this portal. I'll illustrate how the portal provides access to some of these tools, for example, Placa. Tutorials on the website are presented in order to provide instruction about use of the program and configuration of the data set. I'll show you the beginning of the web tutorial for the interface.
Placa. To use the interface, first choose the files you want to upload using the Choose Files button. These files contain early gene expression data under normal conditions and under perturbation of each.
Another tutorial is available for structuring of the data set.
Here I explain the format of the files needed for the web interface of Plato's cave algorithm, also called Placa. These files contain early gene expression data under normal conditions.
In addition, example files for testing the algorithm and a downloadable MATLAB package for Placa are available via the web portal. Using the sample file, I'll show how Placa works. You choose the file, upload the file. This is an experiment in which microarrays were measured with inhibitors of various components of the cell signaling network, and then run the program. Then the program displays the inferred network from the data. By clicking on any of the arrows, the genes involved in inferring the functional component are displayed, and the experimental results obtained from the inhibition data of any of the components are also graphically displayed by clicking on the component. This is typical of how we have designed our software tools so that they can be downloaded and executable for the sophisticated users and are fully documented both with video tutorials and written tutorials for the less experienced user, and they can be operated via an easy to interact with web interface.